Good morning, dear students. Today we are going to see the citric acid cycle. This cycle is associated again with the metabolism of sugar, and once the sugar has been metabolized from the glycolysis, it generates pyruvate, and then the pyruvate is funneled into the citric acid cycle. Since this is a cycle, the initial product of this particular cycle will ultimately run and become the final product, wherein the cyclic form of the cycle can be easily calculated or can be seen. So it is also called as Krebs cycle or tricarboxylic acid cycle. Is the hub of metabolic system. It accounts for majority of carbohydrates, fatty acids and amino acids oxidation. It also accounts for majority of generation of these compounds and others as well. Amphibolic acts both catabolically and anabolically. For example, 3-NAD plus FAD plus GDP plus PI acetyl coenzyme A, it gives 3-NADH, FAD, GTP coenzyme and 2 CO2. So this is the overall reaction that comes in play, wherein both the catabolic and anabolic reactions can be seen. If you see the overall overview of this citric acid cycle or the TCA cycle or the Krebs cycle, then it starts with glycogen. From glucose is generated. Glycolysis pathway, we have already gone through this. And when it comes in terms of pyruvate, this pyruvate again is generated, or it generates acetyl coenzyme. This is the major product inside the pyruvate or after the pyruvate, which is funneled in the TCA cycle. So in a cyclic manner, sorry, this acetyl coenzyme A has been worked out and maximum energy has been conserved. Now, the citric acid cycles, all the enzymes that are associated are generated in the mitochondria. Now, where in the mitochondria? If you see the mitochondria very clearly, see here, the inner surface, maximally inside this inner surface, you can see the reactions that takes place inside the mitochondria. Now, if you see the overall structure of the citric acid cycle, C2, correct? So two molecules of carbon, they get converted, they get attached with to carbon C14 compound, C4 compound, and ultimately gives C6 compounds. Then this cyclic reactions takes place, and during the cyclic reactions, there are high energy compounds that are being liberated, which are utilized inside the electron transport chain. So 3 hydride ions, hence electrons are transferred to 3 molecules of nicotinamide dinucleotide, whereas one pair of hydrogen atoms are transferred to one molecule of FAD. The function of citric acid cycle is harvesting of high energy electrons from carbon fuels. See here, from citric acid cycles to oxidative phosphorylation, ultimately 11 ATP are being generated if the acetyl coenzyme A runs through the citric acid cycle. Oxygen is required for citric acid cycle indirectly as much as in electron transport. At the end of the electron transport chain is to generate NAD and FAD. Now let us start the reactions. The first reaction is formation of citrate. Citric acid cycle begins with condensation of 4 carbon unit oxaloacetate and 2 carbon unit the acetyl coenzyme A. Oxaloacetate reacts with acetyl coenzyme A and H2O to yield citrate and coenzyme A. This reaction, which is an aldol condensation followed by hydrolysis, is catalyzed by citrate synthase. So this is the first reaction wherein there will be condensation of oxaloacetate along with acetyl coenzyme A, which is oxaloacetate is a 4 carbon compound, acetyl coenzyme is a 2 carbon compound molecule, and ultimately it generates the citrate that has been formed with the help of enzyme citrate synthase. See here, oxaloacetate with acetyl coenzyme, this is citryl-CoA that is being generated, which is quite unstable, and with the help of this CoA molecule which has been relieved, it is now becoming citrate. Step second, citrate is isomerized into isocitrate to enable the 6 carbon unit to undergo oxidative decarboxylation. The isomerization of citrate is accomplished by a degradation step followed by hydration step. This results in an interchange of hydrogen atoms and hydroxyl group. The enzyme catalyzing both steps is called as aconitase because cis-aconitase is an intermediate. So formation of isocitrate, this is a reversible kind of reaction, citrate and isocitrate, because a metabolic block can be developed, and based on this metabolic block, the reaction can be reverted back to citrate as well. So this is one of the important point that has to be taken into consideration. Aconitase is an iron sulfur protein or non-hemoprotein. It contains 4 iron atoms that are not incorporated apart from heme group. See it, citrate, cis-aconitate and isocitrate. You can see all these reactions are reversible reactions. It means excess amount of isocitrate will be generated, then that can be again been converted back to citrate as well. The poison fluoroacetate is toxic because fluoroacetate coenzyme A condenses oxaloacetate to form fluorocitrate, which inhibits the aconitase, causing the citrate to accumulate. So this is the metabolic block that is being generated. The mode of inhibition is suicidal inhibition. See here, citrate, same again. Next step that comes in place, formation of alpha-ketoglutarate. Isocitrate undergoes degradation catalyzed by associated dehydrogenase to form initially oxalosuccinate, which remains enzyme bound and undergoes decarboxylation of alpha-ketoglutarate. The decarboxylation requires Mg++ and Mn++ ions, and these are three isoenzymes of citrate dehydrogenase. One of which uses NAD, is found in mitochondria. The other two are NADH, NADPH and found in mitochondria. So this is now the cycle have started. We have seen step 1, step 2 and step 3. So in the next lecture, we will continue with the step 3 part, that is the formation of alpha-ketoglutarate, and once alpha-Kg has been generated, then it will be funneled or it will be moved further to another reaction of the carbon compound, which is the 6 carbon compound now reduced to carbon 5 compound. So that's all for today's lecture.